DNA, deoxyribose nucleic acid. DNA has 3 jobs: storing information, copying information, and transmitting information. DNA is a nucleic acid. The nucleic acid is composed of subunits called nucleotides, which are made of 3 parts. The first part is the phosphate group, and the second part is the 5-carbon sugar, and the third part is the nitrogenous base, which has 4 different types: the adenine, the guanine, the thymine, and the cytosine. The phosphates and the sugar both form the double helix. Base pairings are very important when it comes to DNA. Wrong base pairings will cause genetic mutations. Adenine always has to be paired with thymine, and guanine always has to be paired with cytosine. It's like pieces of a puzzle. Here's a visual example of a DNA double helix. The outside is made of phosphate and the 5-carbon sugar. They make up the double helix. Phosphate has a negative charge. The 5-carbon sugars are what hold the base pairings, but the phosphate is in between them. The 5-carbon sugars are deoxyribose. And then in between is where the base pairings go: A to T, G to C, or C to G, T to A. Here's a closer look at the base pairings. Certain base pairings only fit with the same base pairing. It's kind of like a puzzle, only certain pieces fit with other pieces. You can have any combination of base pairings, and it just makes different nucleic acids. They just continue down the line. Here's a closer look at the 5-carbon sugars, which make up the double helix. They have 5 points, like a pentagon. Strands run from the 5' to the 3'. The 5 point is always on the outside, and then on the other side they run the opposite direction because it's complementary but flipped so that the base pairings can match up. On this side, the 3' is always on the outside. The one that starts out 5' to 3' is the superior, and the other one is the lagging. Phosphates connect sugars together. They go in between the sugars, and the sugars hold the base pairings together. Here's a review of the different structures in DNA. The phosphate and the sugars make up the helix. And on the inside, the base pairings go together, and that's thymine and adenine, guanine, and cytosine. DNA replication. Step 1. Helicase unzips the DNA from the origin, and the bases now serve as template strands. Step 2. Primase, which is a small piece of RNA, serves as a primer, so later the DNA polymers can add the new nucleotides. Step 3. DNA polymers adds new nucleotides and makes sure the sequence is correct. It proofreads everything to make sure that the base pairings go together and that the new string is compatible with the old string. The DNA polymers brings in the new nucleotides, which have to be complementary to the old ones. So A has to go T, G has to go with C. So here's the new string that's going with the old one. And in this picture, the old one is the 5' and the new one is the 3'. And the DNA polymers can only work 5' to 3', which means that the primase for the lagging string has to work extra hard. The 5' is always a leading string, the 3' is always a lagging string, because the lagging string takes longer and takes more primer. Sometimes on the lagging string, the DNA polymers doesn't fill in all of the gaps, so you have Okazaki fragments. This is when the enzyme ligase comes and has to be the glue that fills in the gaps so that the old strand can connect with the new strand. Now that all these steps have been completed, you have 2 brand new strands of DNA, or helicases, and the 2 new helicases both have 1 new strand and 1 old strand, and they are complementary with each other. Thanks for watching.